Okay, so I see some yeses and nos. I was thinking, I almost typed, if a transposable element inserts itself in the promoter, will it change the phenotype? And then I thought that question actually is not really fair because we don't know if it will. So then I changed the question to, can it? And then the answer to that, yes, it may. So that will be a yes. And yours, not an even split now, but if you got the wrong answer here, then you're going to learn something from this slide. So then I'm kind of happy that if you got it wrong, because now you're going to dispel that belief. Okay, if the transposon inserts itself in the promoter region, then what happens is it can decrease how much mRNA is made out of this sequence. So in a future slide, I'll show you how does this thing affect how much of this gets made. It will be in the same model, or it will be in a similar thing. It's also going to be this gene. Okay, so will it affect? Yes, it may. And I'll show you what happens in this specific case. And then this other one is another variant. So all of these are different alleles for the chalcone synthase and the upstream and downstream sequences. The one that's called Nivea is the wild type. The one that's called REC has a promoter upstream. And the one that's called Benny has not promoter. It has a transposon in the promoter. And the last one has a transposon in the promoter and a transposon downstream. And you might say, who cares if it's downstream? Because what regulates transcription is this part. So if transcription ends here, they forgot the little poly A here. This is from a paper. Okay, if its transposon is downstream, can it affect transcription? So don't worry, I'll show you the answers in the end, but let me see what